But mostly what I'm going to show you then is possibilities. And the emphasis is going to be on sort of detecting cell migration. And last night you saw these wonderful movies, mostly from your group, yes?
No, it's Mike Daly.
Mike Daly's group. Okay. Where these activated microglial cells will move to the injury. And we've also heard today from the SPEC perspective that sort of watching cell migration has not been that productive clinically. And I think the problem is if you look at many cells and where they're going, you maybe don't add much, but if you can watch individuals move, you learn a lot. And I think all of the new optical microscopy tools, the two-photon microscopy and being able to label cells genetically specifically are just beginning to show that we barely understand how immune cells move in most tissues, how they interact with their surroundings. And so the goal of the work I'll show you is to be able to someday be able to make the same kind of movies you saw last night, but in an MRI scanner, so that I can watch cells move in your brain or your peripheral nervous system. No? Good. So the focus is going to be on MRI for me. Let's see, where's the button? Oh, I'm upside down.
That's the dead one.
Oh, that's fine. There it is. And the reason MRI is because of the way MRI makes images, you can get this spectacular resolution. Right now, spectacular for radiological techniques, terrible for light microscopy techniques. But here's now your typical millimeter resolution image of the brain. And not only can we make images at high resolution, but the contrast is spectacular, this showing the white and gray matter. Sometimes contrast due to the tissue properties, and sometimes due to added agents. In this case, a little bit of gadolinium chelate to the blood has gotten into the brain. Normally it wouldn't, thanks to the blood-brain barrier, and that enhancement is due to this MS lesion. MS known to be a autoimmune disorder, an inflammatory component. And you know, this result has been remarkable. 30 years later, MRI transformed the way physicians could diagnose MS. It's monitored the time course of MS to unbelievable specificity, which has led to the ability to do clinical trials on much fewer patients. And so I think now it's just a huge impact on the monitoring the progression of MS. So this is, for me, the gold standard. This is what we'd love for every disease, an ability to diagnose, to monitor the progress, and to make it easy to do clinical trials. So MS is the paradigm, but stroke is not far behind now. And stroke is a nice place to illustrate that the MRI contrast is very, very rich indeed. This is work from my colleagues, Larry Latour and Steve Work, where we have a scanner right in the local emergency room at Suburban Hospital. So if you've had a stroke there, they make this blood flow measurement by watching how fast GAD moves, and you can see the blood flow is slower. Brighter means slower in this part of the brain. They weight the image to the diffusion of water, and if tissue is damaged, water will diffuse slower, and so it looks brighter. In this case, the diffusion is bigger than the perfusion deficit is bigger than the diffusion deficit. That means dissolve the clot with TPA. So here's the image right after TPA was given to this person, and you can see the perfusion deficit's gone. The tissue is beginning to salvage, that is, the diffusion anomaly is getting smaller. And then a week later, when you look at the chronic infarct, it's quite small. Had there not been TPA, it would have been at least this large, if not had grown. So this is a case where MRI is being used sort of in real time to make therapeutic decisions and to follow the efficacy of the therapy. So, another wonderful standard to try and achieve. And the nice thing about all of our technology is things are just constantly improving. I show here a couple of images from my colleague Jeff Dunn. These are very high-resolution images, about 200 micron, in plane off of our 7 Tesla MRI scanner. And if you look, what you can see are these cytoarchitectural features that Vince was after. This is the stripe of Generi, the myelin stripe. that runs through the visual cortex. Here's a section through the motor cortex. Again, the scale bar here is 5 millimeters and 10 millimeters. And what you can see here is the transformation as the motor cortex turns into the somatosensory cortex. And so if you were a histologist, you would draw a cytoarchitectural boundary right about there to distinguish the motor cortex from the somatosensory cortex. What's nice about this is the person's still alive. So we think this is going to bring in sort of a new era for anatomy by MRI. But what I want to talk about is are we going to be able to watch cells move, and especially few cells down to the single cell level? And the sort of two targets are, first, we'd love to watch immune trafficking in the brain. And while the brain is immune privileged, the immune system still gets into and talks to the brain. Lymphocytes are entering the brain, dendritic cells are entering the brain, although hardly any in a normal brain. And who knows what they're doing in most disease cases, except things like MS, where we know there's huge infiltration of lymphocytes. And they have preferred routes of entry through the arachnoid space, through the choroid plexus. Where they go, how they traffic, how long do they surveil, very little information about that. So that's one group of cells we'd like to track. And I'll end showing you where we are on that. And the other group of cells we'd like to track are these new neurons that are born in your brain constantly, especially in the hippocampus. And from the subventricular zone, cells will migrate to the olfactory bulb. And these are, again, very sparse. Here's a stain of one of the layers of the olfactory bulb, the glomerular layer, and here's one of these new neurons that were born and positioned themselves right outside this glomeruli. They're very sparse. There's a lot of indication that they're important for the health of neural circuits, but exactly why and how they're working it has been a challenge, and we think being able to see them in intact brains would be interesting. Simple problems like if you change the odor environment of a rat, do the number of cells migrating into the bulb change? Rochefort here would say yes. If you enrich, you get more cells with a rostral caudal gradient. However, that's not been a robust result. McGavin et al. Would say no, they can't see any difference in cells in the bulb with odor enrichment. In one case, they fractionated the bulb. In the other case, they didn't. They just looked at the whole bulb. Now, how are we going to see single cells? So I'm going to claim I'm seeing single cells. Don't be confused. This is not like the pictures you saw last night, where with 1 micron resolution, I could see a cell. I'm going to play a trick. I'm going to put enough MR contrast into the cell to make it look much bigger than it is. And the agent we're going to use is iron, any magnetic material. Iron oxide is the most popular. And that's because a little bit of iron oxide changes the magnetic field because it's magnetic. And MRI is wonderful at measuring magnetic fields to about 10 parts per billion. So if there's just a very small change in magnetic field, we can tell something has disturbed it. So if you take sort of a micron worth of iron, you will see shifts in magnetic fields up to 150 microns. Small shifts, but shifts that we can detect. So the trick to seeing single cells is not resolving the cells, but getting enough contrast so that they bloom large so we can tell they've been there. So cell tracking by MRI is become a robust preclinical field, and a handful of papers where this has been done in humans. And two strategies have been dominated. Number one is add iron oxide formulations. They tend to be dextran coated. They tend to be 20 to 80 nanometer size particles. And if there's macrophages that have infiltrated a tissue, they'll gobble up those iron oxide from the blood and you see them. So here's a normal kidney. Here's a kidney that had just a few minutes of ischemia, and a day later the iron oxide was given, and you can see this dark ring, which shows that macrophages have infiltrated this kidney and eaten up the iron oxide formulation. Every time, I'm going to show you images where the cells will look dark. It's just the way we do it. We can invert the scale or use MR tricks to make them look bright as well. Remarkable to me is that the amount of ischemia was very short. but enough to bring in the macrophages into this region right between the cortex and the medulla of the kidney, which is known to be very sensitive to ischemia. And so this sort of experiment's been done in just about every condition in an animal where macrophages are known to infiltrate. It's quite a robust experiment. The other class of experiment, and I think the early work by Matthias Hohn at the Max Planck and Joe Frank at the NIH were the best examples of this, where you have cells in a culture dish, you play some trick to make them eat a lot of iron, and then you inject them back into the brain. And Matthias did this with embryonic stem cells in a stroke model. And so here you can see these two injection sites, dark holes where a lot of cells were injected. And then over time, you can sort of see the cells begin to migrate out from the injection site and will even form around the stroke area. And with the rise of cell therapies, or the interest in cell therapies, the ability to track these cells is, of course, very, very seductive. And again, in animal models, many people labeling up many different types of cells with MR contrast. So now what got us interested is a number of years ago, Steve Dodd, working with Chen Ho, my colleague then at Carnegie Mellon University, were labeling lymphocytes with these iron oxide particles. And this shows you a lymphocyte with some vacuole filled with these iron oxide particles. And based on their results, I did a back-of-the-envelope calculation that said we should be able to see single cells. And Steve went on to show that. So here's a tube filled of dilute cells. And even more dilute cells. This is just in a tube. And you see these dark spots, and that corresponds to the single cells. So it's possible to get enough iron into the cells to see single cells. These are the tubes with no cells in them. But you had to play hard tricks to get enough of these nanoparticles in. You needed a few million. So Eric Shapiro in my lab decided, well, instead of using the nanoparticles, why don't we use micron-sized particles? So we call these MPIOs. And it turns out these particles used quite frequently by cell biologists to separate cells. So these are just standard particles you'd get from Bangs or Dynel, and you might use them to magnetically sort cells. And cells just love to take them up. So here's a hepatocyte with the 1.6 micron. Eric went up to 10 microns. Here's 5.8 micron particles where they look like bowling balls. And being able to label just about every cell type we've tried to label. Here's mesenchymal stem cells and prostate cancer cells. And so the nice thing about this is 1 micron particle is 1 million nanoparticles. And so just getting a couple of particles means we've got enough iron to see the cells quite robustly. And one of our first favorite examples of this is Cathy Kelly at the NIH. Likes to watch prostate tumor cells metastasize to the brain and treat them. And she was able to, after a few weeks, count tumors. So here in the MRI, 5 weeks later, you can see tumors that arose from these prostate cells that she injected IV. But she didn't know whether she was preventing metastasis or growth of tumors with her treatments. And so we just set out to count cells. And so here's just a few days after injection of the cells, and you can see here's a cell labeled with these MPIOs, and you can actually watch this turn into a tumor. I think here's another cell. This one's a little bit slower. The tumor's not growing as large as quickly. But literally, we went around the brain, and here shows a normal brain with no cells, and here you can see the blips in signal intensity due to cells. In histology, here's the cell, in this case, red fluorescent protein. There's our green particle in that cell. And literally, we counted cells, about 150 a day after injection. And within a week, just a few cells remain that will turn into the 15 or 16 tumors that grow in the brain. So this is a case where we're just counting the cells. And she's busy now working on ways to prevent metastasis to the brain as opposed to ways to block tumor growth. In those cases, we put many particles into a cell, so easy to see. But it also turns out if a cell has only one particle, we can see it. So here's a dish of hepatocytes, and Eric Shapiro, again, just sprinkled a few particles. So every once in a while, there's a cell with one particle. And if you look at the MR, different size scale now, you can see that one particle gives us plenty of signal in the MRI to detect. So now, rather than having to get millions or hundreds of particles into a cell, we just have to get one, which means instead of bringing the cells to the particles, we decided to bring the particles to the cells. And this is targeting these new neurons that we want to be able to watch. And so here, this was the work from Goldman and colleagues, I think now at Rochester, was in New York at the time, injected a virus, the dividing cells make fluorescent protein, and over 2 weeks you can watch the cells migrate as long as you cut the brain and looked inside. So what we did is just inject the particles close to where these cells are born in the subventricular zone, and after a couple of weeks you can see the dark contrast migrating out from this ventricular region into the bulb and fanning out into the bulb. Now, where we inject the particles is a big distortion in the field, so the image looks ugly, but it's around here that we really want to watch these particles move. So literally, you can watch the particles move out. So here's the olfactory bulb. Sorry, I've switched directions. The bulb is here. The back of the brain is here. The nose of the rat's here. The tail is here. Inject the particles, and at the beginning, you don't see anything. After week 1, you begin to see some of this contrast migrate out. By week 2, it's hitting the bulb. By week 3, distributing around the bulb. And it's just another animal that shows the same, roughly the same time course. If we kill these cells with RSC, which kills dividing cells, so now you can't make the cells. What you see normally is there's that migration of cells out of the subventricular zone. It's blocked. And this is just the histology to show us that the RSC really worked compared to very few cells along the RMS. This is called the rostral migratory stream, the RMS. Very few cells compared to normal where there's lots of cells. Lots of cells. So this now is labeling an endogenous cell population and watching it migrate with MRI. How do we do? We're not so good yet. We're not labeling 100% of the cells. We only label about a third of the cells, and it's about a third of all the different cells that get made. A third astrocytes, a third are neurons, a third are oligodendrocytes, and many of these cells die along the way, and they'll leave the particles behind in microglia. So a huge limitation for us is we don't know what cell we're in. We don't even know if we're in the cell that we started in. The label is not cell autonomous, and this is all inspiration for the next generation agents. But still useful. And that is, every time we see a spot, we know a cell was there. So here's a 50 micron image. James Sumner's work now through the olfactory bulb, and everywhere you see a dark spot, boom, 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 is a cell that got from the subventricular zone through the rostral migratory stream to the bulb. And James, I talked James into being crazy enough to literally count the cells in normal animals and in animals that had been odor enriched for a couple of weeks. And now not only are we counting cells, but because we can do cytoarchitecture with the MR, we can break the olfactory bulb into its layers, the glomerular cell layer and the mitral cell layer and the external plexiform layer. And so the images of the cells, which here I've inverted so they look white, now turned red, can be overlaid on the anatomy so we can put the cells on the right layer in the olfactory bulb, and the animal is still happy and alive. And lo and behold, what did he find? He agreed with both of those original stories, papers I told you about. If you look at the glomerular layer, there's no significant difference in the number of cells. If you look at the external plexiform layer, there's no significant difference in the number of cells. However, if you look at the mitral layer, there's been a doubling in the number of cells due to the odor enrichment. Now, that for us is a large change, and what's amazing is no one cares about these cells in the mitral layer. The field just doesn't think this is an important place the cells go, and this is where we're seeing all of the effect. Just to show you what it looks like, here's sort of a control. There's this bright part is the mitral layer. Every once in a while, we'll find a cell. If we odor enrich, much easier to find cells right in that layer. If you do the immunohistology, the cells that have particles look a lot like the granule cells, if you know about these new neurons. They're calretinin positive, so there's a new calretinin interneuron with one of our particles in it. And they're tyrosine hydroxylase positive. These interneurons will make dopamine. And so here's blown up now for you, a new Th positive interneuron. This one has a couple of particles in it as well. So we're very excited. We think we can watch the migration, count the cells. 3 minutes, and follow them into the bulb. Okay. I'll do it in 3 minutes. Now, one of the nice features about using micron particles is instead of doing chemistry to make them, we can microfabricate them like Intel. And if we were as good as Intel, we'd make these nano-sized. And so you can shape these particles into various shapes. This shape here, which Gary Szabo made, is very interesting. It's a double disc shape, as is the cylinder. And what's interesting about it is it has a very homogeneous magnetic field in the center here, which shifts the water in very well-defined ways. So in that way, we can now distinguish this disk, you see, which is different looking than this disk. So we can actually make these particles so we can distinguish them over a broad range. And the particles I showed you were terrible. They only have 30% iron. They're not very uniform. When we microfabricate just simple disks, we're 10 times better than the particles I showed. So I think we're going to get order of magnitude improvement in seeing the cells. And so just to show you that we can now distinguish these particles, Gary made these double disks in different sizes, and there's the image of the red disks, there's the image of the green disks, there's the image of the blue disks. So much like fluorescence with these iron oxide particles, we're going to be able to add color to the cell. So you can make one cell one color, another cell another color. So let me just quickly end with the model we're using to begin to track immune cells in the brain. Because I think this is a place where I think there would be a big impact in following disease if we could actually watch specific cells in the blood enter and migrate through the blood. And we're doing this with my colleague Dorian McGavin, who likes this LCMV model, lymphocytic choriomeningitis virus. He's got ways of infecting the brain with this virus. And so here you can see the virus all green, lighting up subarachnoid spaces. And it's well known that many lymphocytes infiltrate to try and clear this virus. Tatiana and Jadzavek is doing the hard job of trying to label lymphocytes. They're very hard to label. But you can see in this imaging flow cytometer that we can get lymphocytes with particles. And we have an assay that distinguishes whether particles are outside the lymphocyte or inside the lymphocyte. So we can sort for lymphocytes where we know we have these micron particles inside the cell. And we can inject them into the brain. So now this is not a live brain. This is a fixed brain. But here's a case where cells were injected right into the brain. Right here, this brain had been infected with virus for about 4 days. And you can see the lymphocytes have migrated around all of the spaces where we know virus is being produced in this brain. It's just a different view of that. So we can put the lymphocytes into the brain and we'll watch them traffic. And the last very preliminary result is here's a case where we took lymphocytes. Again, these are antigen specific. That is, we know these lymphocytes recognize the virus, injected into the blood now, and you can see that the lymphocytes are entering the spaces, again, where we know there's virus, especially these ventricular spaces and the subarachnoid spaces. So we think we're on the verge of being able to take an immune cell, inject it, and follow it. And I think the coming years will be making sort of these nice microscopy movies of cell migration, not over small areas like microscopy does, but over the whole brain. So next I see you, I hope I'll have movies for you of that.
On that very last data set, how many cells did you inject?
Yeah, it's, right now we're not very efficient. So it's about 10 million cells going into the animal.
And that's the image at how many Tesla?
This is a fixed brain at 14 Tesla. At about, more important is the resolution. That one was run at about 50 micron resolution. On the bulb, I showed you most of those images were either 50 or 75 microns, and with these new array detectors, we'll have 50 micron resolution over the whole rat brain. The particles, as I said, are pretty good. They're good out to 200 microns, which is about the resolution we're using on the human brain at 7 Tesla now. So it's all within possibility.
I'm curious, maybe you can explain it to me. on one hand, metallic objects are not allowed during MRI, especially those that are ferromagnetic. And yet, you have those microparticles with iron oxide. Don't they move under magnetic field?
Well, they are used at the right radio frequencies. You can they will
so you're
heat.
Basically oscillating the field so they cannot move too much.
No. It's a good question. So if you suspend a cell in water and put it on a magnet, you will pull the cell down. Yes. Once inside the cell, we've not been able to move a particle. It takes a very large magnetic field gradient to actually move a particle through cytosolic forces, actin, myosin. And we've been trying on other ways to try and move particles in cells.
Even the microcells.
In this case, the field, so it's not the size of the magnetic field that matters, it's the gradient, the size of the magnetic field gradient that matters for force. And these fields are very homogeneous. They're built to be, they're homogeneous to hundredths of a part per million.
I recall that fatal accident with the oxygen tank.
Yes.
Big object.
Yes.
And it killed the patient.
Yes, but that's a huge moment in a magnetic field gradient. There, the moment of the tank is huge, so you get a big force.
Yeah. So
these are very small moments.
But the microparticles would have larger moments than the nanoparticles.
No, but very small moments. Like I said, it's 10 parts per billion shifts in the magnetic field. If I put a metal tank in there, the field would shift. Tesla. Okay, so it's 10 to the minus 12 less force.
Okay, it's really great, really exciting work, and I think it would be great for animal studies, tracking, looking at self-trafficking. What about if you put them in put that in humans? How long do you think the iron particles would get clear from the brain? Because iron is neurotoxic, so you kind of don't want to.
So the dextran particles have been FDA approved. Their on-bottle use has not been very good, so they're not being used. There is already a company forming to cross-link the dextran-coated particles to larger things, so that you need fewer, and they would be degraded. So the dextran-coated are degradable, but I should say these microfabricated particles, we can put anything we want on the surface.
Right.
So they can be titanium-coated.
Right.
So now what will the FDA say compared to a hip implant to a few of these particles inside you? I don't know. We've not yet begun to discuss that.
Because has anyone actually looked at the clearance? Like, do they get clear?
So in our case, the particles I showed you are extremely stable. They're coated with a polymer coat that doesn't degrade. And,
So
and we've
that's
looked out 6 months, and they're still
So
sitting.
it stays there, yeah.
Stays
So
there.
That's what I was concerned about with humans.
Yeah,
that
yeah.
If it stays there,
Yeah.
it could be neurotoxic.
Yeah. Again, I think if coated in an inert material, let's say like titanium, it would be pretty inert. Again, I don't know what the FDA will say about that.
Okay.
And again, we think we'll need very few cells to see, so there will be very low level. You probably have breathed more particulate matter sitting in your body than we'll need to put in. So, but we'll see. I warned you that if it translates, it will take at least as long as Vince's idea.
Simple clinical question. What's the ultimate fate of these particles? Do they just accumulate in tissues forever, or?
Yeah, so I guess there are a class of dextran-coated iron oxide particles that degrade. Our particles are intentionally non-degradable, so they sit. So we've, in the one case where we labeled hepatocytes and counted them move into the liver, 6 months later they were all still sitting there in the hepatocytes that we had transplanted.
The question is, you sort of get one diagnostic shot with these things, and then after that it's just a cumulative process in the body.
Again, many things are going inside people, stents, hips, that are made of materials that we know are inert, and we can manufacture those with the same material. Now, the FDA likes the fact that you can go in and pull that stuff out. And in our case, you may not be able to pull out the particles, but in the end, I think we could make them quite inert and need very few. But I don't know what the FDA will say.